So as I introduced myself this morning, I'll repeat that now. My name is Sarah Crawford. I'm from the USA, Connecticut State University. I have a cancer biology research lab there. And I've been studying cancer for many years in various aspects, including experimental therapeutics, all the way to basic mechanisms that control the molecular decision to begin the journey of neoplastic transformation. Now, today, what I want to talk to you about, I've been interested in brain cancer for a long time and have studied it in various ways. But today, what I want to talk to you about is my most recent project that we are actually just beginning. And it incorporates one of my major interests in cancer biology, which has always been the involvement of the immune system with the development of cancer. And this isn't something that's very accessible in research. You have the tissue culture system where you can't really study the immune system interactions because you're dealing with a single cell line, which is not interacting with the immune system. You can look at animal models, and animal models such as mice, you can inject the tumor and study its properties, but you don't have the native immune system present, and you're injecting a fully formed tumor into the mouse. So you can't ask fundamental and basic questions. Yes. You can ask fundamental and basic questions. If you're studying most forms of human cancer, you also can't really get in there and see what's going on in those very early critical stages where the cellular decision is made to move away from the normal proliferative pathways in development and regulation within the body to this abnormal phenotype. So after thinking about it a very long time, I decided to look into drosophila, the fruit fly. So we're going to be talking about that today. But before I do so, I just want to lay the groundwork here of what some of these issues are as far as we understand cancer today. So I call this the cancer paradox because there are some very basic unanswered questions with respect particularly to the involvement of the immune system. Now, there's been a lot of research currently being done clinically to use various T-cell therapeutics to treat cancer. I'm interested in that, but in terms of more fundamental processes, what is the role of the innate immune system in tumor development or repression, as the case may be, in activating the immune system in such a way that it will prevent the tumor from progressing? And there's been a lot of open-ended questions, and I don't have time in a short talk to really get into all of the nuts and bolts of this. But suffice it to say, we now understand very clearly that the nascent tumor elicits an immune response. And that was not clear for many years, but we know that that is true now for many reasons. So the question is, as we go from normal tissue, the normal arrangement of tissue, to the development of a tumor, how does that happen? And what is the role of the immune system? And if the immune system is involved, why doesn't it suppress tumor development in all cases? Although it may play an important role in many cases where we never get to the clinic. So what is going on? Well, we know all the way over on the left, we know that we can have germline or somatic DNA damage associated with mutations that are involved in proliferative changes, as well as derailment of normal developmental pathways early in life. This can lead to increased cell number and ultimately to tissue disruption in the tissue of origin. And this is really the basis of the first immune signal that's generated. It's not that the tissue is substantively different in terms of its antigenic properties, but that its arrangement is different. Its contacts are different, and those differences transmit a signal to the immune system in the form of the innate immune system. Now, what may happen is that this interaction causes components of the innate immune system to attack and destroy the tumor directly, as well as to initiate the adaptive immune system, which then goes in vis-a-vis T cells, T cell lymphocytes, CD8 lymphocytes, destroying whatever remaining cells are there. But we certainly, and the outcome to this is tumor cell death. But we know that that's not always what happens. What can alternatively happen? And the question is, where do we make this molecular decision? What is its basis? Alternatively, despite the fact that we are activating an innate immune system, which appears increasingly based on new research to play a direct role, as well as the adaptive immune system in destroying the cancer, sometimes what happens is, particularly in the context of cell proliferation abnormalities due to genetic mutations, this becomes an abrogated response. And that responses that normally should result in tumor cell death are now permitted to go in the other direction of further cell proliferation. And that further cell proliferation ultimately results in immune system suppression through multiple means, some of which involve inflammation, dysregulated inflammation initiated by the cancer that no longer goes through the proper channels, but ultimately sends a signal to repress further immune system responses because of the hyperactivation of immune system signaling. Those immune system altered responses also can directly implicate invasion and metastasis, because the signals that are induced and not causing cell death instead facilitate invasion and metastasis. So our cancer paradox is, if the immune system is involved and activated as cancer develops in a nascent form within the body, why is that not sufficient to remove the cancer in many cases? So as I mentioned to you before, this is not something that we can necessarily study in humans. We can't get inside the brain and look at patches of stem cells which may go awry, forming glioblastomas. We can't look at pediatric cancers as they develop, but we can do so in the fly, in the native system with an intact innate immune system. And I don't need to, well, I might, because many of you are clinicians, but I do mean to say that Drosophila has played an incredibly important role in our understanding of genetics, a la T.H. Morgan, development, regulation. It continues to be a phenomenal tool for understanding biological processes. And there is an important precedent in using Drosophila as a model, because many of the same developmental genes and pathways are conserved. And also we find that up to 70% of genes that are involved in human disorders are conserved in Drosophila as well. So this is why I decided to choose the fruit fly. Now, the Drosophila gene that I am using as the basis of the initial genetic change leading to malignancy is called BRAT, which stands for brain tumor. And this particular gene is temperature sensitive, which means that we can turn it on and off in terms of the temperature as to whether or not it's going to be active or inactive. And if you inactivate the gene, that allows for tumor development to occur. So this is a very nice model to start with. In addition, we have in this system the fact that we have only an innate immune system. There is no, in Drosophila, there is no adaptive immune system. Therefore, we can specifically target and inquire about the role of the innate immune system as it responds to the developing tumor. Now, this shows you some photographs of the late stage larval brain, the normal brain, and the brain in the Bratt tumor, where you have the cerebral hemispheres specifically very much enlarged due to hyperproliferation. And this ultimately will result in the death of the fly. And although brain tumors do not generally metastasize due to the anatomy of the organ, if you take some of those cells and transplant them to other parts of the fly, they will metastasize and cause the death of the fly in that way as well. So the Bratt mutated brains are 10 times larger than normal. And we can see that in histological analysis, you can see that there is a change in the transition from early embryonic proliferative progenitor neural cells. I'll just go past this. I think I have the picture I want to show you. This just shows you the staining of the developing larval immune system. But what I wanted to show you is, in this case, we have specific staining of neural cells. And this is in the neurons in the normal brain. And this is the hyperproliferation, the darker region stained in the Bratt mutant, where you don't have the usual sequestration of the regulatory components that are required for differentiation from the early stage neuroblasts, where normally what happens during larval development is that the neuroblast, because of sequestration of Bratt and some of its target proteins, then distinguishes itself into a subclass of cells called ganglion mother cells, which then progress to form neurons. In the absence of Bratt, where there's no sequestration, we just simply continue to clone these proliferative cells, which then develop to form abnormal cells and a tumor. This shows you the gene arrangement. Here we have the overall DNA locus, where we can look at the intron exon arrangement of the genes. And we can see the actual processed transcript containing only the exons, and the functional exon here is 5, which embodies the coding region of the protein. And this protein has approximately 40% homology at the level of DNA sequence to the human gene TRIM3, which is involved in brain cancer. Now, this just shows you the amino acid sequence. And this shows you some of the mutations that have been identified. Some of them are critical mutations that are nonsense mutations that knock out completely the function of the protein. The ones that I'm working with are missense mutations, which render the protein temperature sensitive. Now, in humans, as I just mentioned, there is a Bratt brain tumor ortholog called TRIM. And TRIM has been shown to play a very important role in brain tumor biogenesis by a very similar mechanism. Again, by sequestering regulatory proteins in subcellular products, not subcellular, but cellular products of differentiation, which then cause further neuronal pathway development. In the absence of TRIM, the primordial neuroblasts overproliferate and ultimately form a tumor. And this is seen in about 30% of glioblastomas. So it is a very representative and critical human cancer whose mechanism of origin, as well as the nature of the gene, is highly conserved among these very divergent, in appearance anyway, species. So deletions have been seen in this gene, TRIM3 in humans. They're highly specific to specific forms of GBM and are very much associated with the etiology of this very lethal form of human brain cancer. And this is just a description diagrammatically of the relationship between the different forms of TRIM, human TRIM, and BRAT gene. And we see that in terms of the protein, they have the same structural domains, including beta boxes, coil, and beta propeller. And the TRIM3, which is the human analog most closely related to BRAT, is about 48% homologous to the drosophila gene. And all of the mutations that block the normal functions of this tumor suppressor are found in the beta propeller region. Now, human glioblastoma is one of the most serious forms of human cancer. It currently is not treatable, and glioblastoma itself counts for about 50% of human brain cancers and can occur at all ages. Many of them occur in midlife, but there are some glioblastomas that progress from gliomas in young children as well. And here we see notch signaling involved, where the TRIM protein binds to the notch protein, and that upregulation of notch in the absence of TRIM contributes to the failure of these cells to differentiate properly. And here you can see TRIM3 in its normal configuration. You can see that in the left panel. This is a protein blot. And on the right side, we see in patients, many patients with GBM, there's decreased or absent presence of TRIM. And in the absence of TRIM, notch is no longer regulated and proliferation goes way up. So again, I hope I've successfully convinced you that there is a fair amount of analogy between these two proteins, and that the mutant system in Drosophila represents a legitimate model for studying glioblastoma. But the goal here is not just to study glioblastoma as a singular entity, but to see it in the context of systemic effects, particularly the immune system. And that is my goal here. So as I mentioned before, the absence of adaptive immunity in Drosophila provides us a unique opportunity to study the early stage interactions of the innate immune system with the developing cancer. And this involves pathways such as TOL, JNK, and JAK-STAT, which in Drosophila play a role in its antimicrobial defense mechanisms, which are very important, but also play a role in an anti-cancer effect in developing tumors within the Drosophila as well. And these relationship correlations between TOL, JNK, and JAK-STAT are conserved in humans as well, in the human innate immune system. In fact, a few years ago, Nobel Prize went out because of the discovery of the toll receptor pathway in Drosophila, and its analog in humans was found after that. And of course, the innate immune system, again, I'm not going to get into a great deal of detail in the absence of time, but there is both humoral, as in the human immune system, and cellular immunity that are important components. And that's sort of illustrated here, where the cellular component involves the hemocytes. Over 50% of them ultimately become elaborated to immune system cells, similar to macrophages. And then we also have the humoral component, which involves the production of cytokines and immune activators associated with destruction of the bacterial host. So these pathways have analogy. And what we want to look at then is to see what happens. This is the goal of our research. We are beginning it and taking a look at these early stages and trying to decipher exactly what will be going on here. So we know that the activation of JNK signaling is critical to the response of the innate immune system to the developing tumor. And that this is triggered in part by the breakdown of tissue structure, also compression. There are biomechanical forces, changes in the electrophysiological properties of the cell, as well as mobilization of calcium, which plays a very important role here. All of those differences will trigger the immune system. And they trigger JNK, where JNK then, and this is in Drosophila as well as humans, will trigger JAK-STAT, and JAK-STAT in turn will result in the production of cytokines, as well as activate in the Drosophila, the hemocytes to form plasmatocytes, AKA macrophages, which will then destroy the tumor cells. Now, one critical aspect of this that we're going to be looking at is the fact that one of those key questions in the cancer paradox is if you turn on a growth control gene, an oncogene, you tend to abrogate the immune response. And what is it about that that changes the critical dynamic? And we're going to be looking at that in terms specifically with the tumor-associated hemocytes, which are triggered not only by the development of cancer, but of course by induced by wounds and other infectious disease agents. So going back to this notion, which goes all the way back to Virchow, that cancer is a wound that does not heal. Here we have, again, this notion that there is a basic fundamental abrogation of that healing pathway, which is driven by abnormal cell division, which then generates this invasive. So we go from what would otherwise be a benign tumor, but the aberrant interactions with the innate immune system will trigger the invasive properties. So, and these really are critical. The fact that the immune cells themselves appear to participate in this process because they begin to secrete MMPs, they invade, and they trigger a microenvironment that supports the tumor cells to start behaving like immune system cells gone awry. So we have then the anti-tumor effect specifically of the plasmatocytes, which are a specialized version of the red blood cell, myeloid lineage in Drosophila. They are anti-tumor immune cells. We are studying them in Drosophila in response to the developing brain tumor. They are recruited to the site of the tumor. Their proliferation will be activated, so they will increase in number. They will express tumor necrosis factor, which is also found in humans, as well as IGR, which interacts with IGR to induce tumor cell apoptosis. And this would occur also in response to infection. And the TNF is another, tumor necrosis factor is another critical component of this pathway, but we don't really understand the nuances of their interaction in the context of the nascent tumor. So we're going to be looking at all the levels of these immune system mediators at specific stages of tumorigenesis and to see why and how their levels are not sustained or their functions become altered, and can we intervene in those pathways in order to disrupt the process of nascent tumor development, which is a big goal. And then again, here we see that in cancers, of course, we have this inflammatory activation associated with nuclear factor kappa B, also involved in Drosophila, and this regulatory component. We know in humans, although NF-kappa B is important for regulating immune responses, in tumors, it gets to higher levels where it no longer initiates innate immune system responses, but now triggers a runaway inflammatory response that drives tumor invasion and metastasis. So the abrogated tumor immune system response, is this the molecular basis of metastasis? So we're looking at that with respect to invasion in the developing cancer in the brain. And we can also test it by transplanting that to another portion of the fly to see if metastasis is occurring. We know that based on the research of many other prominent scientists, such as Cordero, showed that hemocyte attachment and infiltration of the tumor will provoke the cells to induce the high levels of IgG expression in the tumor-associated hematocytes, and this should kill off the tumor. But we know that that does not happen. And again, this is that positive feedback loop that is initiated by hyperproliferation, and hyperproliferation itself drives further mutation and selection for a proliferative phenotype. And thank you. And in the context of that happening, this is where the system basically goes out of control and homeostasis can no longer be achieved. So I will finish by coming back to my original question, the cancer paradox. And so the goal here is to take the developing, this is just a generic cartoon really, but in the case of Drosophila, taking specific embryonic, and it's believed that these changes are initiated early in embryogenesis, even before it becomes apparent in Drosophila that a tumor is developing. And that this breakdown of pathways, instead of differentiating, this loss of this gene causes enhanced proliferation. And this goes back to one of the most fundamental issues of cancer. So this is our question. We know that it can run out of control. And what we want to do now is to take apart the immune system responses and to see under what conditions the innate immune system may be able to abrogate the abnormal cell proliferative signals that ultimately, if unchecked, will develop into a tumor that bypasses homeostatic mechanisms and becomes the driving force of the system, leading to its ultimate destruction. So that is the work we are engaged on now. And hopefully I can come back next year if I'm fortunate enough to be invited, and I will tell you how this work is progressing. So thank you so much for listening.